Thank you, Claudio. Good evening, good afternoon to everyone. I am very happy to be involved in the event and very happy that I can share some results with my colleagues and also with students who I believe can take some hints how to proceed with their own experimental adventure. So now I will share my presentation. I hope it is visible. Can you confirm that you can see the presentation?
Yes, Elwira, it is very clear.
Okay, so I will try to be in time. So as you can see, my major interest is metabolomics, but metabolomics as a tool. So it means that we can choose different type of work, but the most important is the aim of this work. It means that we should focus what we would like to obtain firstly before we start any research. It means that, of course, at the beginning of our scientific work, we develop our workshop. So we should learn some skills, how to use procedures, how to use equipment. And finally, when we obtain this knowledge with time, of course, we should ask the question, what we would like to, how we would like to use these tools and what we would like to obtain in the end, because it shouldn't be in that way that we do something and just without thinking produce some results and try to do something with the results. On the contrary, you should put the aim at the end of our journey. At least we should develop an idea for some project and then try step by step realize this project. Of course, everyone gets to the point that something is difficult, something is, let's say, disappointing, but still we should see the aim of our project. So in that case, metabolomics, it is only a tool, as you see, and I focused on discovering the impact of plant metabolites of bacteria. In the other words, I can say that I try to, with my colleagues, with my network of collaborators, try to find the way in which this plant secondary metabolites affect bacteria. So we can say, try to evaluate, discover what is the mechanism of action of this plant secondary metabolites in bacteria and how bacteria respond, also in developing some kind of, let's say, maybe not resistance, but the response on this plant metabolite. So at the beginning, for these colleagues who are not many familiar with metabolomics, mainly who are focused on bioactivity. So metabolomics, it's a general term which involves the work with small metabolites of any kind of a cell. It may be plant cell, it may be bacteria cell, it may be cell of a grower organism like a human cell, but in general, we speak about metabolites, which are quite small. It means less than 1.5 kilodaltons in size. These metabolites can be substrates, this can be different intermediates and also products of the metabolism of the cell. And what does metabolomics do? We can, by using metabolomics, we can just observe the state of a metabolism in the cell in a given time and also in a given environment. So it means that we can just observe by metabolomics. And metabolomics is, of course, compatible and should be, in the wider view, run with proteomics, also transcriptomics or genomics. But here is a focus on metabolomics. What we can obtain, what kind of information we can find with the application of metabolomics for antimicrobial finding. These are two ways. As you can see on the right, it can be selection of antibacterial compounds in the mixture in the plant extract, let's say. So this means usually a simple screening with determination of MIC value in the extract. Also for pure compounds, so we take extract or isolated compounds, check how its performance on the selected bacteria, and say what is the most active. So so in this case we define activity of the plant compounds. On the left we can see that we can try to understand the mode of action of these compounds. So it means that we expose our bacteria, selected organism, with to the plant metabolite, and then what we extract not plant metabolites anymore, but we extract metabolites of bacteria, and we check what was changed in this bacteria after the incubation with a stressing agent. So this both, let's say, approaches lead us also to bioinformatics, which is another tool needed now in the let's say era of computers to process a lot of data which we generate with different techniques. And as I said before, when we proceed in the selection of antibacterial compounds, we can use basic chemometrics or for example, molecular networking. I will not go into details for this to explain these terms, but everyone who is interested can just dig a little bit in the literature, which is very, very let's say, well available and extensive to find about these terms, but this leads us to antimicrobial drug leads finding. When we proceed with the understanding of mode of action of natural compounds, we can explore the target points in bacterial metabolism, and this can be done by a so-called classification strategy. When we have for example, antibiotics, which exert some action in bacteria, and we have unknown compounds. So we treat bacteria with both, and we try to compare if our unknown, let's say, compound with unknown mechanism of action has any similarity to different types of antibiotics. And we also can proceed with pathway analysis, indicated which pathways were altered in the organism, and also with the integration of transcriptomics, genomics, and metabolomics. And similarly, we go for uncovering mechanism of bacterial adaptation and resistance. So we see that we can obtain two different ends of the story when we only check the mechanism of action, but we also can try understand how bacteria react and if bacteria develop any resistance for our compounds. And this can be done also by integration of transcriptomics, genomics, and metabolomics, as well as comparative metabolomics. And this again leads us to antimicrobial, antibacterial drug leads targets in the living cells. A few words about metabolomic peopling, which is applied in our laboratory, and this starts, of course, with the experimental design. Because we are working with Mycobacterium tuberculosis, only a few words about this organism, which is important because Mycobacterium tuberculosis is very slowly growing bacteria. So it means that the cell divides once per 24 hours. So to obtain the biomass, which you can see in the flask, it's around 3 to 4 weeks that the colony forming units start to be visible and start to clot together, and we should wait very patiently for a bigger biomass of bacteria. Why I'm talking about it at this point at the experimental design? Because when we know our let's say advantages which is the advantage of slowly growing mycobacteria, is that we very likely to observe the metabolic changes in this bacteria, because they are not so quick in the metabolism, so we are more probable to obtain any differences. But a disadvantage is that if we plan something badly and we do not obtain the let's say targeted result, we have to repeat everything once more and we lose a lot of time. So it means it is not planning easy to repeat, but we lose not only resources but a lot of time. So experimental design is very essential for, of course, for any kind of research. But if we are dealing with something what is very slow, and this slow procedure is not dependent on our laziness, but on the growth of bacteria of the organism itself. So this is very important. As you can see, of course, a few steps are always required. It means after the incubation, extraction, also we can design what kind of extract we would like to study, if extracellular or intracellular. Again, we end up with the analysis. And what kind of methodology we use for analysis also depends on our experimental design. So when we obtain results, doesn't matter if, at this point if it is LCMS, NMR, or it is FTIR or any other, let's say, tool, technique, we proceed for a computational data processing, because nowadays it is very easy to be supported by bioinformatics, and also to obtain, let's say, statistical normalization of the results. It means that in case of biological replicates, we need at least for metabolomics from usually from 6 to 10 biological replicates. So we can see that to obtain reliable results, we should have some uniformity between our samples. And computational data processing is very useful, because we easily can find which samples are not similar to the others, and we can exclude it. And then we still need statistical data analysis. It means that very often it allows us for a visualization of the data. Of course, visualization is only, again, a tool to help us to interpret the data. But yes, also for determination of statistical significance, and for to observe any changes between the data sets which we can generate. At the end, we have the time for biological interpretation. It means, again, that we have some tools which can allow us to connect our observations. For example, if we are studying metabolites, but also studying genes, we can proceed with connections between genes and metabolites, and draw the pathways which were upregulated or downregulated. Also, enrichment ratio for different biological processes which we studied within cells. As we can see, there is here only a few examples of results. There is huge available number of tools also available without any fee in the internet, through the internet, which we can use to process our data. Only we need some basic skills, some basic training in the how to use this tool. So this is the metabolomic peopling, and what we can do later. I would like to show you a few examples of results obtained through this metabolomic peopling. These are 4 different, let's say, plant metabolites or extracts. And what is the difference between them? The difference was the approach. during the study. So here is an example of cinnamon aldehyde, which was used from cinnamon essential oil. And what we have here is LCMS metabolomics, supported by bioinformatics, by processing, which enabled the determination of upregulated and downregulated compounds within bacteria cells. And underline again, this was only LCMS metabolomics. As you can see, when we treated bacteria with our compound or essential oil, we obtained different clusters. On the left, you can see that it was a cluster of treated bacteria and also of control bacteria. And here cloud plots, which shows us which number of compounds were upregulated and downregulated. So the first observation was the cinnamon aldehyde caused significant alterations, and here we focus on the lipids in mycobacterial cells, but also some metabolic pathways were altered. Few hints about the results. So we observed the changes within the outer membrane of mycobacteria cell. Just one brief word about the schematic here. So we can see that it's a typical scheme of Mycobacterium tuberculosis cell envelope. So we can see the layers: cell membrane, also here peptidoglycan arabinogalactan, the layer of mucolic acids, and and so-called capsules. So these are not covalently linked molecules. And what was changed? What was affected with treatment? We observed changes upregulation in the fatty acids, mucolic acid, glucose monomycolate, and thioteryl dimethylcerosates, which are part of the outer membrane. So this part was thickened by the action of cinnamon aldehyde. Also, we observed increased synthesis of building blocks for lipomannan and lipoaramilomannan, which usually are attached with to fill the holes in the mycobacterial cell wall structure. And this structure also have some immunological function. However, it was not the aspect which we studied. So in case of the other metabolites not related to lipids, we observed the induction of the xenobiotics metabolism. It means that bacteria wanted to overcome the highly reactive species within cell and neutralize the xenobiotic agent which was given them. So the other example, what was changed? We used also NMR metabolomics with the collaboration with the colleagues who I will acknowledge at the end. So you can see that firstly started only with LCMS. We added also approach for NMR at the same, let's say, at the same paper. We compared what we obtained with both approaches. And again, similar slide what you have seen before. So we observed changes. So it means that the selected metabolite was active against bacteria. And what we obtained, also results were connected to the lipids forming cell membrane and outer membrane, upregulated of these lipids. Again, so similar observation for a previous compound. And additionally, we also observed increased flavin biosynthesis involved in the redox reaction and increased arginine biosynthesis, which is usually a defense antioxidant in the cell. And this observation about the arginine and flavins were also obtained from NMR metabolomics. Again, to spread the possibilities, to spread the ideas. So we used for the third metabolites, third extract metabolites for Salvia melitorhiza, with attentioniones produced in the Salvia melitorhiza root. We used LCMS metabolomics and also transcriptomics. With selected genes, we checked expression of selected genes, which code transcription factors needed for response of bacteria to the outer environment. So this time, correlation of metabolomics and let's say transcriptomics. Also, the change was the type of extract obtained, because previously we checked only what was inside bacteria, but here we tried to show what was in the bacterial layers. So you can see that we have 4 graphs, and every graph is from the different type of extract. It was from the extracellular, outer layer, inner bacterial layer, and also inside the cell. And again, here to underline the layers, culture medium, it was upregulation of many molecules, which were usually not observed in the medium, meaning that the tanginions caused some release of this intracellular compounds into the medium, meaning that they acted like let's say surfactants, which disturbed the outermost part of the cell envelope. And the outermost part was extracellular, let's say outer layer. Yes, I'm sorry. Here was inner layer. It means cell plasma membrane. Observation, we had some downregulation here, and also inside cell, also majority of statistically significant compounds were downregulated. Only 1 or 2 compounds were upregulated. And the major observation was the disruption of the bacterial cell envelope because of the release outside the cell of compounds which usually are not placed in the cell culture medium, and also numerous lysoforms of structures which are based on the phosphoinositol. And it is worth to say these lysoforms are produced through the destruction of the full forms. So it means that some kind of catabolism within these forms was upregulated during the treatment. In case of transcription factors, we try to correlate with these factors which were upregulated with their function in the cells to have any, let's say, connection between metabolites and the genes. In case of last example, it was propolis, and for a first view, the same methodology, so LCMS, metabolomics, and transcriptomics. But what was changed? Also different type of extraction and different incubation time. What was important? Firstly, all previously shown experiments were done within 24 hours. It means 1 replication of bacteria cells. And here you can see that also after 48 hours, after 2 days, so we wanted to see if the action of propolis was stable or bacteria overcome this action within time. So as you can see, there were no statistical significance within extract obtained with chloroform and methanol. These are lipophilic extract, usually used to extract lipids. So no statistical significance here. Only methanol, alcohol, methanolic, aqueous methanolic extract were statistically significant, different between groups within control bacteria and treated bacteria. And what was within this extract? These were only amphiphilic lipids. So it means this which has also affinity to more polar solvents, and also the other small metabolites. And as we can see, very tight groups were after first day of incubation, but surprisingly, or maybe not surprisingly, after second day of incubation, still we can see the difference between groups, but bacteria are started to produce the wider variety of these compounds. And the major observation after 24 hours and 48 hours were related to the lipids, amphiphilic lipids, which are part of the cell membrane. As you can see here, after first day, there was upregulation in these lipids, so it means that bacteria started to produce, to seal the cell membrane, and after 24 hours it was like a balance, so it means that bacteria were much, let's say in the common words, feel much comfortable and safe with the propolis within it. However, we obtained a nice interesting observation within small metabolites which were related to riboflavin, but also sulfur-containing amino acids, which is cysteine and methionine. Of course, I'm not going into details here, but it was related to redox homeostasis, related to mycotiol and sulfur metabolism within Mycobacterium tuberculosis. And again, we tried to correlate transcription factors. SIG8 was highly upregulated, and it led us to the identification that redox reactions and metabolism of the sulfur-containing amino acid were regulated also not directly but indirectly through SIG8, and we observed this changes through metabolomics. So at the end, this is the last slide. I would like to share the message to you that, as you can see, this is a network of transcription factors defined for Mycobacterium tuberculosis, but also this is a network of our collaborations. As you can see, here is Department of Natural Product Chemistry, Medical University of Lublin. I work in this department, but also here is the Department of Microbiology of National Institute of Tuberculosis and Lung Diseases in Poland. This is a reservoir for virulent strains for me, which are not available in my university because of safety reasons and lab facilities reason, and also Stefan Engelov's Institute of Microbiology from Bulgarian Academy of Sciences. This is a contribution of Professor Milan Georgiev and his team, who was supported with metabolomics and also some bioinformatics, and also Department of Biochemistry and Biotechnology, my colleagues from Medical University of Lublin. So as you can see, any work which we try to design, which we try to develop, which network of people working together. So home take message is to develop some network for you, and also to try to work with people, to understand each other, to discuss a lot, and then you can obtain your goal which you settled at the beginning. So this is my message for you. Thank you.
Dear Elwira, really congratulations. It has been a very interesting and excellent presentation, and also you have been also really punctual, because you finished it perfectly at 15 and 5 minutes. So we have minutes for any question that participant would like to ask to Professor Elwira Seniawska. Please, the discussion is open. Obviously, PhD students are warmly invited to ask questions to Professor Seniawska.
I would like to pose a question while all other people are shy, so we take advantage of your presence here. And yeah, I want just to ask you your opinion about the role of the scientific information, the rational information that can derive applying the metabolomic approach and the network pharmacology, not on a single compound, rather than interpret, at least in part, the effect of an extract, of a complex extract. With a specific approach, of course, you should know at least the main compound to interpret with the network pharmacology. Do you think that this approach can be useful or rational to drive further investigation in vitro, in vivo, and so on, experimental investigation that, of course, will be necessary to interpret the activity of an extract?
Yes, of course, I agree with you, Luigi, that it is very important and nowadays, let's say, more famous approach, because we need holistic approach to understand not only what is a drug, but also what is the base of the disease. I agree that network pharmacology can provide us some kind of solution, because we have tools. When we produce a network of associated factors which are affected by the extract, we also can use bioinformatic tools to calculate which compounds in the extract are most probable to be active. However, from the applicational point of view, there is still a big, let's say, reserve for the extract. Why? Because we cannot be sure which compound is active for sure until we isolate it. So I think it's worth to go deeper in this direction. But however, we still have to be aware about the limitations if we are going to the final products, because at the end, at the market, they will say, no, it's not the drug, it's only the extract, let's say supplement or something. But within cell, we have to prove that which compounds are active, but not discussing the metabolites in vivo, because it is a totally other different topic.